Hello friends, this is Nilanjana and you are watching BioStudy Pathshala. So through this title and thumbnail, you have known about today's topic. So don't waste much time and let's get started about today's session. So let's learn duplicate recessive epistasis. So duplicate recessive epistasis. First, what is the meaning of epistasis? It means to mask. Now we are going to draw two homologous chromosomes. So these are two homologous chromosomes. On these two chromosomes, in this particular loci, A gene is present. And as it is homologous chromosome of this chromosome, so on this loci, the variant of A gene will be present. For example, here the recessive allele of A gene is present. And on this loci, for example, B gene is present. And here again the variant of B gene will be present. For example, the recessive allele of B gene is present. In case of dominant and recessive epistasis, the epistatic gene masks the effect of hypostatic gene's expression. So on these two videos, we have learned that A was epistatic gene and masked the effect of B gene's expression. In case of recessive epistasis, the homozygous recessive form of A gene masked the effect of the B gene's expression. And in case of dominant epistasis, the homozygous dominant condition of A gene or heterozygous condition of A gene masked the effect of the B gene's expression. So this was about the recessive epistasis and dominant epistasis. Now what is about the duplicate recessive epistasis? Here A gene products, these are capital A, capital A small a and small a, and B gene products, these are capital B, capital B small b and small b. So the homozygous recessive form of A gene and homozygous recessive form of B gene will mask each other. That means the homozygous recessive form of A gene will mask the effect of capital B or capital B small b, and the small b will mask the effect of either capital A or capital A small a. So if on a particular genotype homozygous recessive condition of A gene is present, then it will mask the effect of the entire genotype expression. And if the homozygous recessive form of B gene is present, then it will again mask the effect of entire genotype's expression. That means if small a, capital B or small a, capital B small b, these two genotypes will show same type of phenotype, because here homozygous recessive form of A gene will mask the effect of these genotypes. Again capital A, small b and capital A dash or capital A, small b will show same type of phenotype, and here small b will mask the effect of these two genotypes, and it is duplicate epistasis. So here two genes, these are A and B gene, interact with each other, and the phenotype of A and B gene will be same phenotype. That means the A and B gene code for same characteristics or same result. For example, A gene will result formation of red coloration. That means B gene will follow the same pathway to give rise the red coloration, and the genotype of A gene that are capital A or capital A small a will result the red coloration, and the homozygous dominant condition of B gene and the heterozygous condition of B gene will lead to formation of red coloration. But the homozygous recessive form of A gene, that is small a, and the homozygous recessive form of B gene will show same type of epistatic event. That means these two genotypes will mask the effect of these other genes to give rise a particular phenotype. So this is all about the hypothetical example. Now the actual example is the flower color. The flower color of sweet pea. The flower color of sweet pea follows the duplicate recessive epistasis. The sweet pea scientific name is Lathyrus odoratum. Lathyrus odoratum. So this plant's flowers show two types of colors. These are purple and white, and in between these two colors, white is much more recessive than purple color, and these two colors are results of two genes. These are C and P genes, and these two genes will lead to formation of purple color when one dominant allele of C gene along with one dominant allele of P gene is present. That means two dominant allele of each gene, that is C gene and P gene, is required to formation of purple coloration. If small c, capital P or small c, capital P is present, then it will lead to formation of white coloration. Again capital C, small p or capital C, small p is present, then it will lead to formation of again white color. And the last genotype, that is small c, small p will lead to formation of white color. So now we will see about the cross procedure of this sweet pea plant to know about this duplicate recessive epistasis phenotypic ratio. So let's learn. So in P1 generation, a white colored flower crossed with a white colored flower. Now this white colored flower show the genotype as capital C, small p. That means white coloration. And this individual will show its genotype as small c, capital P. That means again white colored flower. So these two individuals will produce 4 gametes in each individual, and this will be known as gametic 1 generation. So in gametic 1 generation, this individual will produce capital C, small p type of gamete. Obviously the number of gametes will be 4, and this individual will produce small c, capital P type of gamete, and the number of the gametes will be again 4. So these gametes will fuse together to form F1 individual. So in F1 generation, the individuals will be the purple flower with genotype capital C, capital P. That is purple phenotype. So now we will see the selfing between F1 individual, which further lead to formation of F2 individual, and this selfing will be occurred in between two purple flower. So this purple flower with genotype capital C, capital P, the purple flower crossed with a purple flower which has the genotype as capital C, capital P, which is purple flower. Now this individual will produce 4 types of gametes. These are capital C, capital P, capital C, small c, capital P, and small c, small p. This individual will produce 4 types of gametes again. So first one is capital C, capital P, capital C, small c, capital P, and small c, small p. Now we will see the F2 individuals through checkerboard. So this is the checkerboard of F2 individual. Okay, so these are the F2 individuals, and in this line the gametes are male gametes, and in this line the gametes are female gametes. And here total 16 individuals are obtained. You can see through the color differentiation, these individuals are showing purple coloration. That means 9 individuals among 16 are showing the purple coloration. Now in this individuals, first one is showing homozygous dominant condition of C gene and homozygous dominant condition of P gene. Here you can see homozygous dominant condition of C gene and heterozygous condition of C gene. It is the same, and in this the heterozygous condition of C gene and homozygous dominant condition of P gene, it is the same of this one, and this is the heterozygous condition of C gene and heterozygous condition of P gene. These four are with the same genotype, and from this data we can say that one dominant C gene or one dominant C allele with one dominant P allele results in formation of purple color. So C dash, that is capital C dash, capital P dash, results in formation of purple coloration, and with this particular genotype the number of the individual is 9 among 16. You can see here as well. Now here capital C, small p, small p. Here you can see also capital C, small p, small p. That means homozygous dominant condition of C gene with homozygous recessive condition of P gene, or heterozygous condition of C gene with the homozygous recessive condition of P gene. This is again the same genotype, will lead to formation of white color. So from this data we can write down in this way that capital C dash small p will lead to formation of white color. So capital C dash small p lead to formation of white color, and with this particular genotype the number of the individuals are 3 among 16 individuals. Here you can see that homozygous recessive form of C gene with heterozygous condition of P gene. This is the same genotype of this one, and this is the homozygous recessive form of C gene with homozygous dominant condition of P gene. It will be homozygous dominant condition of P gene because here the gamete is small c capital P and small c capital P. These two gamete fuse together to form this genotype. So this genotype is again showing the white coloration. So from this we can write down in this way that small c with one capital P or one dominant allele of P gene leads to formation of white coloration. So the small c capital P dash leads to formation of white coloration, and with this particular genotype the number of the individual are 3 among 16, and the remaining last one, that is small c small p, all the homozygous recessive form of 2 genes lead to formation of white color, and with this particular genotype the number of the individual is only 1. So from this data we can say that 9 among 16 individuals are showing purple coloration, and this 3 genotypes leads to formation of 7 out of 16 individuals which show the white coloration. So from this checkerboard we can obtain that the phenotypic ratio as 9 purple is to 7 white. That means the phenotypic ratio in case of duplicate recessive epistasis is 9 is to 7. Although I have forgotten to tell something new, that is the duplicate recessive epistasis is also known as complementary gene interaction. It is also known as complementary gene interaction. Okay. So we have learned about the duplicate recessive epistasis cross procedure, the phenotypic ratio of the duplicate recessive epistasis, and the example which follows this pathway. Now let's learn about the enzymatic pathway for duplicate recessive epistasis. So now we will see about the enzymatic pathway for duplicate recessive epistasis. In this pathway the basic color is white, which is coded by small c, small p. That means all homozygous recessive form of C and P gene lead to formation of white color. Now this white color modified to again white color. This is intermediate color, which is coded by capital C dash small p, and this transition occurs due to presence of E1 enzyme. This E1 enzyme modifies this white color with this particular genotype to this white color with this particular genotype. Now this E1 enzyme is the product of capital C or capital C small c. That means homozygous dominant condition of C gene or heterozygous condition of C gene lead to formation of E1 enzyme, which further modifies this white color with this genotype to this white color with this particular genotype. Now this white color further modified to purple color, which is coded by capital C dash capital P dash, and this modification occurs due to presence of E2 enzyme. This E2 enzyme transform the white color to purple color, and this E2 enzyme is the product of capital P or capital P small p. That means the P genotype product is E2 enzyme, which lead to transformation of white color to purple color. So this is the pathway for duplicate recessive epistasis. Now if instead of capital C or capital C small c, if homozygous recessive form of C gene is present, it will lead to formation of no functional E1 enzyme. If it produce any E1 enzyme, it will be non-functional. So this non-functional E1 enzyme will lead to formation of white color, which will be coded by small c, small p. Now if a functional E2 is present, it will lead to formation of this white color to again white color, which will be coded by small c, capital P dash. This E2 enzyme is product of capital P or capital P small p. The precursor of E2 enzyme is this white color, which is coded by this particular genotype. So E2 enzyme only can act on this white color or only on this genotype. It cannot act on this genotype. So here, although the functional E2 enzyme is present, but it cannot show its effect because here the genotype is small c, small p. Now if homozygous recessive condition of P gene is present, then it lead to formation of non-functional E2 enzyme. That means again here the E2 enzyme cannot act. So this non-functional E2 enzyme lead to formation of white color, but this white color will be coded by capital C dash small p. That means although one dominant allele of C gene is present, but the homozygous recessive allele or homozygous recessive condition of P gene suppress the pathway. So from this pathway we can say that the homozygous recessive form of C gene suppress this pathway and homozygous recessive condition of P gene suppress this pathway. If the intermediate white color, which is coded by capital C dash small p is present, if here again small p is present, it will lead to formation of white color because it cannot transform this white color to purple color, as the condition is small p, that means non-functional E2 enzyme. Even if E1 enzyme is non-functional, that means if the genotype is this one, then also the E2 enzyme cannot show its effect on this particular genotype, which will lead to formation of again white coloration coded by small c, capital P dash. So in our video we have learnt about duplicate recessive epistasis, the cross procedure, the phenotypic ratio, the example and the enzymatic pathway for this particular gene interaction. So if you have learned something through this video, do like this video, share and recommend this channel to your friends, and also do subscribe if you haven't till now. And if you have any queries regarding any lectures or anything to know, you may let me know on comment section, as well as you can message me on mail ID, business mail ID given on description box below. So till then, thank you.